Okay, so remember that we have a quiz on Wednesday over the urinary system. And so these are the two different types of nephrons. So which one is the one that has the long loop of Henle? Does anybody remember what that one's called?
I don't remember.
Juxt.
Oh,
Juxta, right? So the juxtamedullary nephron. So if you remember that, the juxtamedullary nephron is unique in that it has this long loop of Henle that extends down into the medulla. And then it has these special capillaries, which are called the vasa recta, which function in the reabsorption of water. So just as a little review for the function of the nephron, I'm going to show you a little animation video of the nephron. It's about 5 minutes. And then we'll specifically talk about the reabsorption of water and how that can be altered depending upon whether you drink a lot and you're well hydrated or whether or not you're dehydrated. Let's see if I can get the volume to come on.
The veins and the urinary tract. The kidneys are paired, bean-shaped organs lying outside the peritoneal cavity in the back
Help
of
if
the
I moved it
upper
over there, wouldn't it?
one on each side of the vertebral column. The kidneys function as the body's main excretory organs, eliminating the body's metabolic waste products by filtering the blood. Substances that are unneeded or are present in excess are filtered out of the blood and form into urine, moving via the ureter into the bladder before being expelled through the urethra. The kidneys also selectively reabsorb those substances that are needed to maintain the normal composition of the blood. By adjusting blood composition, the kidneys are able to maintain blood volume and pressure, ensure the proper balance of sodium, chloride, potassium, calcium, hydrogen, phosphate, and pH, and eliminate products of metabolism such as urea, uric acid, and creatinine. The medial border of the kidney is indented by a deep fissure called the hilux, where the blood vessels, nerves, and ureters connect to the kidney. The kidney is composed of up to 18 lobes. Each lobe is composed of nephrons. Nephrons are the functional units of the kidney, and each kidney contains more than 1 million nephrons. Each nephron consists of a glomerulus and a system of tubules. The glomerulus is a unique high-pressure mass of capillaries that filters the blood. The glomerulus is encased in a thin, double-walled capsule called Bowman's capsule. The space inside the capsule and surrounding the glomerulus is called Bowman's space. Plasma-like fluid is filtered from the capillary blood into Bowman's space through the glomerular filtration membrane. The glomerular filtration membrane consists of 3 layers of capillary wall: the endothelium, the basement membrane, and the epithelium. This membrane allows some particles from the blood to pass through, but not all. The fluid that is filtered from the capillary blood into the Bowman's space is called filtrate and forms the primary urine. The filtrate then diffuses across Bowman's space and into the tubule system of the nephron. In the tubules, some substances are added to the filtrate as part of the urine formation, and some substances are reabsorbed out of the filtrate and back into the blood. The nephron tubule is divided into 4 segments. The filtrate passes through each of these segments before reaching the ureter, a highly coiled segment called the proximal convoluted tubule, which drains Bowman's capsule and where almost complete absorption of nutritionally important substances takes place. A thin loop structure called the loop of Henle, which reabsorbs water and ions from the urine and plays a role in controlling the concentration of urine. A distal coiled portion called the distal convoluted tubule, which regulates potassium, sodium, and pH, and where further dilution of the urine takes place. And the collecting tubule, which joins with several tubules to collect the filtrate and where final sodium regulation takes place. Each kidney is supplied with blood by a single renal artery that arises on its respective side of the aorta, before dividing into 5 segmental arteries that enter the hilux. Within the kidney, each segmental artery branches into several lobular arteries. The lobular arteries further subdivide to form interlobular arteries, which branch off into afferent arterioles. Blood flows into the glomeruli through the afferent arterioles. Blood flows out of the glomerulus through the efferent arterioles. The afferent and efferent arterioles regulate glomerular capillary pressure by selectively dilating or constricting. The kidney's venous blood, now filtered, flows from the glomerulus via the afferent arterioles into the peritubular capillary network, a low-pressure, reabsorptive system surrounding all portions of the tubules. This arrangement permits rapid movement of solutes and water between the fluid in the tubular lumen and the blood in the capillaries. The peritubular capillaries rejoin to form the venous channels by which blood leaves the kidneys and empties into the inferior vena cava. Urine formation involves the filtration of the blood by the glomerulus to form an ultrafiltrate of urine, the tubular reabsorption of electrolytes and nutrients needed to maintain the constancy of the internal environment, and the secretion of waste materials. Filtration occurs as blood flows into the glomerulus from its afferent arteriole, and plasma moves through the glomerular capillaries into Bowman's space. From Bowman's space, the glomerular filtrate moves into the tubular segments of the nephron. Here, through tubular reabsorption, electrolytes and nutrients move from the filtrate back into the bloodstream. Here also, through tubular secretion, substances move from the peritubular capillaries into the urine filtrate. The filtrate concentrates in the collecting tubules, then finds its way to the renal pelvis, where it is directed to the ureter, the bladder, and the urethra for elimination. The kidneys perform an excretory function by filtering the blood and then selectively reabsorbing those materials that are needed to maintain a stable internal environment. The nephron is the functional unit of the kidney and is composed of a glomerulus, which filters the blood, and a tubular component, where necessary substances are reabsorbed into the bloodstream and unneeded materials are secreted into the tubular filtrate for elimination in urine.
Okay. So one of the things that we talked about was the reabsorption of water. And so remember that in the proximal convoluted tubule, we have lots of reabsorption of water, as well as in the loop of Henle. So if we look at this water reabsorption, we have two different types of water reabsorption that takes place in the nephron. And so we have one type of water reabsorption, which we already talked about, which is called obligatory H2O reabsorption. And that means that it has to happen, right? So this must occur. And the reason why it must occur is that water just moves via osmosis. So water moves via osmosis across the surface of the cells. So we know that it has to always happen because water is, the plasma membrane of the cells is permeable to water, so water will just flow out, right? And so this takes place in the PCT, right? And also in the descending limb of the loop of Henle. And we talked about how it is that increasing solute concentration in the medulla that actually draws water out, right? And so this would be passive, right? So it's passive. And the other thing that's interesting about it is that it does not take energy. And that's really important, specifically when we talk about that in the loop of Henle, because what is taking energy is the creation of that solute concentration by pumping sodium out. So remember that sodium gets pumped out in the ascending limb, and then that creates this difference in the solute concentration that will cause water to just passively move out, okay? So water reabsorption is obligatory, but then we also have facultative water reabsorption. And so facultative means that it occurs sometimes, but not always. So it can be regulated, right? And so this is how we regulate H2O concentration in the urine. So this is how we determine whether our urine is going to be dilute versus concentrated. And so this is due to the presence of proteins which are called aquaporins. So these are proteins in the plasma membrane that allow for water to move out of the filtrate. So they're essentially water channels. So just think that they're water pores. And they can either be present, so they can actually be inserted and then removed from the plasma membrane. So there is a hormone that determines whether or not the aquaporins are present. And these are actually found, not the hormone, but the aquaporins are actually found in the collecting ducts primarily. So it's in the collecting ducts that we have facultative water reabsorption. A little bit takes place in the distal convoluted tubule, but I'm just going to talk about the collecting ducts. Okay. So when we look at the hormone is called the antidiuretic hormone. And we talked about this a little bit because ADH is a hormone that is produced by the hypothalamus and is stored in the posterior pituitary. And ADH, when it is present, it causes water to be reabsorbed. So when present, aquaporins are inserted and water is reabsorbed by the collecting ducts. So that is going to make it so that we have concentrated urine. So if you think about what this word means, antidiuretic, when you think about diuresis, diuresis is the production of urine. So antidiuretic would mean that you produce less urine, right? So you produce volume-wise because you're reabsorbing the water, but your urine is more concentrated, right? So antidiuretic would be decreasing the output, the volume of the urine, but it's increasing the concentration. So we produce concentration, the increase in the concentration of the urine, and we decrease output.
Do you do that when you want to get your blood pressure up?
Yes, right. So you can, if your blood pressure is too high, you can take diuretics. Right? And then that actually causes you to get rid of extra water. Antidiuretic hormone would actually increase your blood pressure.
Yeah.
Yeah. Okay, so let's look at a diagram that shows this from your book. So when we look at what happens if we are overhydrated, okay? So this means that we've taken in too much water and we need to get rid of it, some of it, by passing it into the urine. And so we have the ability to measure the osmolality of the extracellular fluids. And so what that is the solute concentration. So if there's a decrease in the solute concentration, so this is osmolality, this means that there's a decrease in the solute concentration, right? This decrease in osmolality is detected and ADH is released by the pituitary, posterior pituitary. Wait a minute, yeah, oh, sorry. If we have a decrease, ADH is not released. You can put not released. Okay. So in this particular instance, notice how there is no facultative water reabsorption. So the water, the aquaporins are not present in the plasma membrane. However, we also have the ability in the collecting ducts to recycle urea. And so we can call this urea recycling. And this also takes place in the collecting ducts. And so what that does is that this causes urea is reabsorbed, and then it causes the solute concentration in the medulla to increase. So it increases the solute concentration of the medulla, so the interstitial fluid in the medulla. So urea, as well as sodium and chloride, are important solutes in that process. Okay, and so notice how urea is passive, right? So urea just passively moves across that membrane. So it must be at a higher concentration inside than outside, and so it diffuses with its concentration gradient into the medulla. Okay, so we recycle some of that urea. Okay, so the other thing that happens is if we are so dehydrated that we need to reabsorb some water. And so look at what happens now is in the collecting ducts, we have all of this water reabsorption taking place. And so that is due to aquaporins being inserted into the membrane. So if we have an increase in the osmolality, which again is just a measure of solute concentration, this causes ADH to be released. Aquaporins, so you can put dehydration up here. Aquaporins are inserted in the membrane, right? And water is drawn out of the collecting duct. And so that is going to concentrate your urine. And so notice again that it doesn't take energy. Okay, so every time you see the blue, that means that it's just passive. Okay. And then we have urea recycling too.
Is urea recycling in terms of like too much water, or is it in terms of
Well, it actually occurs all the time. But in this case with ADH, there's an increase in the urea recycling. So we can put increase urea recycling.
Is it a decrease with if you have too much water?
No, it's just because so it would be higher when you're dehydrated and lower, but it doesn't stop. So it's lower when you are overhydrated. Okay.
Can we stay on it for just?
Yes. So this is an example. What is this? Is this obligatory or facultative? Water reabsorption, right here. Facultative, right? Yes. It only happens sometimes. Perfect. Okay, right here, it's obligatory. It has to happen, and that's just because it's moving across the plasma membrane. Okay. So sometimes in medicine, you'll hear people talking about renal clearance. And renal clearance is actually a measure of what we've kind of already talked about related to glomerular filtration rate. So it's used to estimate. So used to estimate GFR, which is the glomerular filtration rate. And so remember that if that is too low, then that means that your kidneys are starting to fail, right? And if it's so low, then there's no clearance of stuff in the blood. So this is where blood gets, in the plasma gets removed. So this is the amount of time it takes to remove a substance from the plasma.
Is this the test they do with at the hospital to determine if someone's renal?
Yes. Okay. So one of the things that they can do is that they can put something in the blood plasma that can be excreted by the urine, and then they can see how much over a given amount of time comes out in the urine. So how fast it leaves from the plasma and goes into the urine. And so they use oftentimes a chemical or organic molecule called inulin. And so inulin is a polysaccharide that's found in plants that we cannot break up down. Right? So this is put into the blood plasma. Right? So for example, if you put 1 milligram, and there's a description of this in your book, into the blood plasma, and the clearance would be the flow rate or the concentration of the substance in the urine versus the substance in the blood plasma. So we could say that U is the concentration. So this is the concentration. U equals the concentration in the blood plasma. V equals, there's a formula here that I'll show you in a minute, the flow rate of urine formation. So this is in milliliters per minute, generally. And then we get P, which is the concentration of the substance. Oops, did I screw that up? Sorry, U is the concentration of the substance in the, I can't read my notes without my glasses, sorry. So U is the concentration in the urine, sorry, we'll put this in the urine. P is the concentration of substance in the plasma. That makes it more sense. So think P plasma, think U urine, and V is the flow. Okay. Plasma. Okay.
Up at the top where it says polysaccharide, what does it say after?
In plants, sorry, in plants. So it's not something that we would normally have in our blood plasma, right? And it's not something, but it's something that we can excrete. So it's something that we can take out. So if we put a certain amount in, then we can measure how much comes out in a given period of time. So the renal clearance rate, which is C, so C is the renal clearance rate, is equal to U times V over P. So if you just plug in those values, and we kind of use this 1 milligram, so the volume flow rate would be 1 times 125 divided by 1 would equal, so this is just an example, would equal 125 milliliters per minute. So if the amount of in the plasma and in the urine is the same, so it's 1, right? 1 milligram per milliliter, and then this would be the volume of the blood flow, then we get 125, okay? So what this means is that this means that when we're putting it in, right, all of it is coming back out because it's a 1-to-1 ratio, right? So what happens if you reabsorb stuff, then this is going to become smaller. So if we find that substances are reabsorbed, this will become smaller. And if substances are secreted, then this will become larger, okay? So the significance of this is what is normal. So this is normal renal clearance, right? This is kind of how fast it should occur. So I measured, if this is kind of a measure of GFR, if this is less than, so if C is less than 60, right, milliliters per minute, then this is a sign of renal disease. And then if C is less than 15 milliliters per minute, this is a sign of renal failure. Right? So inulin is just something that they put in and they try to estimate how much. So if like only 60 came out, right, that would mean that there's something wrong with the kidneys, that they weren't able to get it out, right, fast enough. It wasn't able to clear the urinary system, or actually clear the blood plasma and get into the urine.
What does this have to do? Sorry, I'd like to. So what we do is we also monitor their output, and usually they have like a certain amount, like 30 mLs per hour is considered adequate, or like just on base. What does that have to do in terms of, because you see like 125 mLs a minute, or like 60 mLs.
So you said 30 mils of urine per hour.
They like 200 every 4 hours.
Right. Well, this is specifically looking at the rate of clearing a particular substance from the blood plasma. So I'm not sure how that directly relates to the amount of urine that they want outputted in a given amount of time, because you can store it in the bladder. And so it's a little bit iffy, because now we're looking at how it's stored in the bladder rather than just looking at the concentration of the substance in solution. Yeah. Yeah. Okay. So one other thing besides inulin that you can actually use in the body is you can actually also use creatine. So sometimes instead of putting inulin into the blood plasma, they just use creatine. So sometimes the clearance of creatine is monitored. And creatine is a form of nitrogenous waste, or creatinine. Okay, so instead of inulin, you could also measure the clearance of that. Okay, so let's look at examples of causes of renal disease. And so when we look at renal disease, some things that happen, or let's look at the causes first. So causes. So in today's culture, the major cause of renal disease is what? What do you think? In our culture, what is the major cause of renal failure? Nope.
No?
Nope.
Oh yeah, you're right.
Nope. Liver, kidney, all that can also do kidney. Nope.
Traumatic injury.
Oh,
Okay, so not insipidus, because insipidus has to do with the regulation of ADH, right? The production of ADH. So diabetes mellitus, right? And remember, we talked about why it's called mellitus. Does anybody remember what mellitus means? Sugar, right? So it's actually honey. So that means that they have sugar in their urine, right? Because they have so much in their blood plasma that they cannot reabsorb all of it from the filtrate, and so it ends up in the urine, right? So diabetes mellitus is a major problem, primarily because it also affects high blood pressure, right? So it also affects your blood pressure, because if you have lots of glucose in your blood, you're also going to reabsorb lots of water to decrease the solute concentrations, and so that's going to boost your blood volume, and that is going to give you hypertension. So we'll also put hypertension here as it is related. Right? But also hypertension can also occur beyond somebody having diabetes, right? So these can both cause damage. You can also get infections, and there is a word for this, which is called pyelonephritis. nephritis. So this is a kidney infection. Pyelonephritis. And so we'll talk about why kidney or how kidney infections can happen in a little bit. They can be caused by urinary tract infections that get up into the kidney. And then we can also have renal causes of, we can also have some drugs. So some drugs can also affect the functioning of the kidney. So with some drugs. Okay. So if we look at the symptoms, one of the symptoms is that you get a buildup of nitrogenous waste in the blood. And the nitrogenous wastes include urea, uric acid, and creatinine. Okay, so those are examples of nitrogenous wastes. And then you also get acidity, so you get a decrease in the blood pH. And when you have kidney failure, sometimes this is called uremia. So this literally means urine in the blood. So what does the nitrogenous waste in the blood pH, how does it affect the body? So it makes people really fatigue easily. They tend to lose weight, right? They have muscle cramping because their ion concentration in their interstitial fluid is not being regulated. And then they also have mental disorders, right? So sometimes they get dementia-like symptoms because of kidney failure, okay? So one way that you can treat kidney failure is through dialysis. And so dialysis is where you actually remove the blood and you transport this blood into a machine that has a dialysis membrane. So remove the blood, right, and put it into the dialysis membrane, which we actually used in lab when we were looking at osmosis in lab, right? And then what happens is that urea, right, and other solutes leave the blood passively. So when you go through that dialysis membrane, the dialysis membrane is actually bathed in a fluid that is actually going to draw the urea out, right? So it actually causes urea to come out, and then some other solutes will come out, and then they can also put glucose back in. So like if a person is malnourished, they can actually put glucose back into the blood there as well. And then they put the blood back into the patient. Right. So they're simulating, by using this dialysis membrane, they're simulating the work that is normally carried out by the nephrons in the kidney.
So it's essentially just recycling.
Yeah, so they're putting it through the dialysis machine, and that just draws out all the bad stuff, and then they can put good stuff back into it passively by glucose can just kind of move back through the membrane, and then depending upon the membrane that they use, and then they can put it back into the patient. So it looks something like this, right? So blood is drawn from the artery, and then it goes into a dialysis machine, and that's a membrane, and this membrane is surrounded by this dialysis fluid. And so the fluid is at a lower concentration, so there would be less urea here, And then urea would just simply diffuse out across that plasma membrane. And so the dialysis machine has to keep pumping new dialysis fluid into and then removing the old dialysis fluid. So they're just using simple diffusion, and because obviously it would be impossible to simulate active transport, right? So they're just using simple diffusion to remove stuff from the blood.
That's that nitrogenous waste.
Yeah, yeah, so the nitrogenous waste they specifically want to remove. Okay. So let's look at the next structure, which is the bladder. So you remember that the ureters enter the bladder, and they actually enter the bladder really low on the bladder. So you think that the ureters would enter it on top, but they actually enter it down low. So enter the bladder inferiorly, right? And this is where we store, so this is the storage of urine. The bladder is primarily composed of smooth muscle. And this actually has kind of a funny name. I think it's called the detrusor muscle. If I can spell it right. Detrusor muscle. So you can think of like thrusting out urine when this particular muscle contracts. So let me make sure I spelled that right.
That would cause the bladder spasm?
Yes, that would cause the bladder to spasm. You know, and what, I don't have that written down. Does that look right to you guys from lab?
Yeah.
Okay. Okay. So the smooth muscle is involuntary, and we actually have also a sphincter, which is associated with the bladder. So there's actually a sphincter, which is called the internal. My notes here, sorry. The internal urethral sphincter. So what a sphincter is a muscle that is circular. So we've actually already looked at sphincters when we were talking about the mouth, right? That would be a sphincter. When we talk about the pyloric valve, that is a sphincter. When we talked about the ileocecal valve, that would also be a sphincter. And the internal urethral sphincter is, and both of these are under involuntary control because they are smooth muscle. So this is smooth muscle. So when the bladder fills up, there are in the walls of the bladder stretch receptors. So the stretch receptors in the wall of the bladder give you information about how full your bladder is. So the stretch receptors can send a signal into the autonomic nervous system, and the autonomic nervous system can actually cause the contraction of the bladder and then the opening of this internal urethral sphincter. So I'll just can send a signal to the CNS, the central nervous system, and the ANS can respond, the autonomic nervous system. So it is actually the parasympathetic nervous system that causes urine to be released. So the parasympathetic division, remember, is not the fight or flight division, but it's actually the conservation of energy and relaxation. So the parasympathetic division causes an increase in urination, and this is called, has a fancy word for it, is micturition. So this is urination. Okay. So in babies, this is reflexive, right? As soon as their bladder fills to a certain point, there's a reflex that goes to the spinal cord, the central nervous system, and then it sends it out, and the muscle contracts and they urinate, right? But once you start to potty train, the really important idea here is that you can override the desire of the body to contract that muscle. And this is done by the external urethral sphincter. So this actually prevents urination consciously. And this is skeletal muscle. Right? So potty training is learning to overcome the urge to urinate, right? The involuntary urination by controlling that external urethral sphincter. Okay. So you can sense this, like, oh, I need to pee really bad. And what is happening is that your bladder is starting to contract, right? And that you have to pee really bad. That internal urethral sphincter opens up because that's involuntary. And then you have to use your external urethral sphincter to not urinate. And then after a while, the bladder will stop contracting, right? So it comes in urges when you need to pee, right? It's like, oh, I need to pee, but then if I just wait a few minutes, then I will stop thinking about it. And then that's when the bladder stops contracting. Right? And so what we see here when we look at this right here is where, this is the male, this is right here is where the ureters enter. And then this is your first urethral sphincter right here. So that's the internal urethral sphincter. And this is also called the urethral orifice where it comes out. And then right here is the external urethral sphincter, which is located right there, right? And so that is actually in the body wall. So that is your perineum. So that is the muscle that is between your anus and your reproductive structures. And so sometimes when people have incontinence, it's because this muscle has become weak, right? So sometimes incontinence is where sometimes urine will come out, right? And in the female, it's the same thing, except that it's a separate opening from the reproductive. Here's your ureters. Here's your urethral internal. Here's your external. This is your external urethral sphincter, right? And so there are exercises that you can do to prevent urine from seeping out, right? And so those are sometimes referred to as Kegels, right? So if you contract the wall, that wall of muscle, then that is going to increase the strength of this and actually prevent urine from coming out. Does that make sense to everybody? Okay. Okay. So if we look at another interesting difference between the males and the females, right? The males, the urethra comes out through the penis. And so generally there is very little way that we can get infection up into the urethral, into the urethra. But in females, the urethral opening is really close to the vagina, right? And it's also really close to the anus. And so when we talk about UTIs, these are urinary tract infections. Right. So normally the urine is sterile. So it does not contain good bacteria or bad bacteria, right? It doesn't have any bacteria in it, right? It is also normally slightly acidic. So pH of urine is usually about a 6. So it's a little bit acidic. It can be more acidic. It can be like a 4, right? Or it can be more basic. It could be like an 8. So it can range from like 4 to 8 in the pH scale. Okay. So the urinary tract infections are more common in females. And they can get into the bladder and they can cause bladder infections. And so from the bladder, they can easily, relatively easily, if it's not treated, they can then get up into the ureters and they can go to the kidney, right? So what you want to avoid is having a bladder infection that spreads to the kidneys, because that can actually damage the kidneys so bad that they'll start to shut down, right? And so you want to prevent infection from spreading to the kidneys. So that's what they typically give you antibiotics for, right? Is to prevent that. And the way that they test for it is they have you give a urine sample, and you can kind of tell if the urine sample, if you pee into a cup and the urine sample is kind of white, and if it's opaque, right? If it looks like it's kind of, there's cloudy stuff in there, then that suggests that there's an infection, right? Rather than clear. So this causes cloudy urine, right? So you could have bacteria and also white blood cells in there. Okay, so we're gonna move on now, and we're going to go to reproductive. So we're gonna start on the reproductive system today. And we're gonna start with male reproductive structures. So the reproductive system, the main point of this obviously is to produce egg and sperm in males and females, right? Egg or sperm. And then also to eventually produce offspring. So we can think about that when we think about the reproductive structures in the male. So we call the testes, these are called the gonads. And the gonads are where we're producing sperm. So this is the production of sperm primarily. Talk about what else it does. And the testes in mammals are located in the scrotal sacs. They're not in other species. So like even in birds, right, the testes is up in the body cavity, even and they're warm-blooded, but even like in reptiles and fish and all other types of organisms, they do not have the testes outside the body. So we have to ask, why in mammals? So I'll just put mammals only. Okay. So the main reason to have the testes in the scrotal sac is that the production of sperm has to take place at a lower temperature. So this decreases the temperature, so it produces a lower temperature than the body, the core body temperature. So like where the ovaries are. And so it's about a -3 degrees Celsius difference in body temperature. And for some reason, that is really important in sperm production. So if the temperature is too high, the sperm are deformed, right? If the temperature is too low, then sperm production shuts down, right? And so that temperature, the maintenance of temperature is important. Now the scrotal sacs can actually be raised and lowered by muscles. And so we have a muscle that is called the cremaster muscle. So we have muscles associated with the scrotal sac. So we have what is called the cremaster muscle. And this is a skeletal muscle that can elevate the testes. So if the testes get too cold, right, for sperm production to occur, then the cremaster muscle contracts and it pulls the testes up closer to the body. It can also relax. So if the testes are too hot, right, it moves them away from the body. So the cremaster muscle also is important in temperature regulation. There's actually two muscles. The other muscle is the dartus muscle. And the dartus muscle is actually located in the skin, and it is smooth muscle. And it causes the skin of the scrotal sac to wrinkle. So if the testes is too cold, the skin will, or that muscle will contract and the skin will wrinkle, right? And then it'll relax. And so smooth muscle, both of these, skeletal muscle and smooth muscle, are relatively involuntary, right? So it occurs without having to voluntarily do it. Okay, so those are the scrotal sacs. Now, connected the testes to the rest of the body, we have what is referred to as the spermatic cord. Spermatic. And I know I didn't spell that right, it doesn't look right. Spermatic cord. I'm losing it. Spermatic cord, that doesn't look right. Let me see if I can find it in my notes here on the images. Spermatic, it's just two words. Okay, got it. Spermatic cord. Okay. Okay. I've completely gone off coffee and I'm just like, okay. Spermatic cord. Okay. So this connects the testes to the body cavity. So this is where the muscles run, right? This also contains the nerves. This contains the blood vessels and the lymphatic vessels. Right, so the spermatic cord is what connects the testes to the body cavity. And it also contains the vas deferens. And we'll talk about the vas deferens, but the vas deferens is what transports the sperm out of the testes and up into the body cavity. So the sperm are produced in the testes, but they actually have to come up into the body cavity before they are ejaculated out through the penis. And so when we have individuals getting a vasectomy, it's important to realize that they do not cut the entire spermatic cord, right? This is just cutting the vas deferens in the spermatic cord, right? And so this prevents sperm from being in the seminal fluids. Prevents sperm from being in semen. Okay. So think vasectomy. Ectomy means to cut, and so that's just cutting of the vas deferens. Okay, so let's look at a diagram of this. Okay, so this is my spermatic cord coming down. That shows the blood vessels and the nerves, right? And then this shows the testes. And we're going to talk about what's inside the testes in a minute. Now, the blood vessels and the nerves that are in the spermatic cord, there's a really important blood vessel that is called the pampiniform plexus. And this is a group of vessels that actually help to regulate the temperature of the blood that is entering the testes. So this removes heat from the blood entering the testes. Right? So you can see that there's a plexus, which means that it's branching, right? So see those blood vessels? Why isn't it just one, right? One of the, like one blue. There's one red going in Why isn't there just one glue coming out, right? That's the penpeniform plexus. And so it helps to remove heat from the arterial blood that is coming in, and that also helps to regulate our temperature of the testes. Okay, so if we look at the testes itself, inside the testes, we have a set of tubules. So if we just look at this part right here, we have what are called the seminiferous tubules. So the seminiferous tubules right here are actually where sperm are produced. So this is the location of sperm production. Now, surrounding the testes is another group of tubules. So this is the structure right here surrounding the testes. And that is where we store the sperm prior to ejaculation. And so that structure is called the epididymis. So this is where we store sperm prior to ejaculation. And it's kind of interesting because we've discovered that sperm do have a shelf life, right? If they're stored too long, they become what is called sticky sperm. And they're, at least in a petri dish, they're not able to move very well, right? They get really slow, and they're not able to fertilize the egg very well. And so sticky sperm is a consequence of storing the sperm too long in the epididymis. Okay. Okay, so then we also talked about the vas deferens. So I'll write that one here as a structure. And the vas deferens carries sperm upon ejaculation. So we'll put upon ejaculation. Sperm is transported back into the body cavity. Okay, so when they label the vas deferens, oftentimes that's the only structure they're showing, but remember the vas deferens runs within the spermatic cord. So if we look at a diagram that shows that structure of the vas deferens, right, this is it right here. So this is the testes. So on the quiz on Wednesday, you might need to identify this. This is the testes, this is the scrotal sac, right? This is the epididymis. The epididymis is the series of tubules that surrounds that. And then this is the vas deferens coming up, right? But remember that it's traveling in that spermatic cord. So it comes up, and so only sperm are found in the vas deferens initially, in the ejaculate initially, but it has to pick up some supplies, right? So it has to pick up some substances. And so we can look at accessory glands in the male. And the first accessory gland is called the seminal vesicle. And there's actually two of them. So there's two. There's only one prostate gland, but there are two seminal vesicles. And so they're shown right here, right? The seminal vesicles. So they contribute substances to the ejaculate. And one of the things that they contribute, so they contribute is nutrients. And the nutrients that's contribute. The nutrients is in the form of fructose. Right? And that is kind of strange because this is like the only place that I know of that we actually use fructose in the body, right? Because fructose is actually fruit sugar, right? It's a little bit different than in the structure than glucose. So we use glucose. So it's like, well, why are they using glucose? But the sperm use fructose, right? And so they're able to break down and use that as energy. So this would be energy for the sperm. Okay. They also contribute a coagulating enzyme. And so when you think about coagulating, you want to think about thickening, right? So what this does is this actually thickens, and it's going to thicken the sperm upon entry into the vagina. So the function of this is this thickens the semen, all the stuff that is being ejaculated, including the sperm, upon entry into the vagina. And the reason for this is that this prevents a phenomenon that is called backflow. So this prevents backflow. So the semen could backflow out of the vagina, right? The female stands up, semen could come out, right? So this thickens, right, and helps to keep it from coming back out. So that's the coagulating enzyme. And then another thing that's kind of interesting that's found is, I'll move over here because I'm running out of room, are what are called prostaglandins. And we have prostaglandins all over our body, but in the semen, it specifically has a specific function. And this actually causes the female's reproductive tract to contract.
Is it still under the seminal vesicles?
Yeah, so this is under seminal vesicles. So it causes the female's reproductive tract, like the vagina and the uterus. So just put vagina and uterus. To contract. And so what this does is this helps move the semen up further into the reproductive system. So this moves the semen up into the reproductive system. Okay, so those are some things that the seminal vesicle contributes to the semen. And I have in my notes that it contributes 60% of the volume. So we'll put 60% of volume. Okay, so that's the seminal vesicle. So you can see here, this is the seminal vesicle. And then it turns into a different duct, which we'll talk about in a minute, but then it goes through what is called the prostate gland. So the prostate gland has a different function. There's only one of them, right? So it contributes about 33% of the volume of the semen. Okay. It contributes another nutrient that is called citrate. And so this is a nutrient needed by the sperm. And then it also produces anticoagulating enzymes. So these would act after the sperm has been in the vagina for a while. So this actually makes the semen become less thick, right? So the semen becomes less viscous, less thick. So the sperm can move. Right, so first you coagulate and then you anticoagulate the semen. They also produce in the prostate gland, and this is kind of an aside because it doesn't have a specific function reproductively, but we have what is called a prostate specific antigen. So this is a PSA. right? So when would your PSA levels be of concern if you're a male? Does anybody know, like, when you would have your PSA levels checked? Yep, so then if you're at a concern for prostate cancer, right? So this is a detection of prostate cancer. So it's used to detect cancer, right? So for some reason, the prostate gland is really, really susceptible to cancer. And so they estimate, like, by the time you're 80, there's, like, I don't know, like a 70% chance if you're male that you will get cancer, prostate cancer, right? And so one of the symptoms of prostate cancer is the inability to urinate. And that's because the urethra actually travels through the prostate gland. And so if you have high PSA levels, that is of a concern, right? If you get prostate cancer, then they just continually monitor the PSA levels. And if it goes down, it's good. And if they go up, it's bad. So that's just something else that the prostate gland does produce, right? That they are able to measure. Okay, now there is another gland that is called the bulbal urethral gland. And this produces what is called pre-seminal fluid. So pre-seminal means it's the fluid that comes out prior to ejaculation. So this would be before ejaculation. Okay. So in the males, this is important because it helps to eliminate urine from the urethra. The urine could be very acidic, and it could actually damage the sperm as the sperm travels through the urethra and not the penis, right? So it eliminates urine from the urethra, and it also functions to lubricate the penis. Generally, it does not contain sperm, although there's some concern in terms of pregnancy if it would. So like if you were thinking about using a natural rhythm or removing prior to ejaculation, sometimes this is of concern that there could be some residual sperm left in the urethra. And so the pre-seminal fluid could have some sperm in it if there were like multiple ejaculations in a given period of time. So if we look at the diagram here, this is my bulbal urethral gland. So here's my prostate gland, right? Here's my seminal vesicles, and then this structure right here is the bulbal urethral gland. And so prior to ejaculation, it is what releases the substance that comes out through the penis, and that just lubricates it and also prepares it for the ejaculate traveling through it. Okay, so I'm going to stop there for today, and then we'll talk more about male reproductive and hormones, and then we'll get onto the female reproductive system.